Hello, my name is Brianna Kozlowski, and for my Organic Chemistry Honors Option project, I decided to research the organic chemistry of luminol. So I will be discussing the reaction between luminol and blood, luminol's importance to forensic scientists, and the properties of the luminol molecule. Luminol is important to forensic scientists in everyday life. The reason I chose to research luminol is because I aim to be a forensic scientist in the future, so I would be using luminol on a regular basis, and we need to understand how it works. Luminol is a solution created by mixing luminol, hydrogen peroxide, and a hydroxide salt in a spray bottle. The most important property of luminol is that it is chemiluminescent, which means that it produces light upon reaction with the iron in blood. This reaction is what allows hidden blood to be seen at crime scenes, since the luminol will allow blood to glow a bright blue. The structure of luminol is important because it determines its function of chemiluminescence. On the structure shown, you can see that there are protons bonded to the nitrogen atoms. These protons give luminol its acidic properties and allows it to react with the base in the solution. This results in a negative charge, which is transferred from the nitrogen atoms to the oxygen atoms. This step allows the oxygen from the hydrogen peroxide to interact with the molecule, which replaces the nitrogen atoms with the oxygen atoms. This reaction allows photons to be emitted, and this allows light to be seen by the human eye. It is essential that this reaction occurs because the end result is what is important to forensic scientists. This process is what allows minuscule amounts of blood at crime scenes to be observed. Without that initial acidic property, the entire preceding reaction would not be able to occur, and the light would never be emitted. Functional groups of luminol include amine, carbonyl, and alkene, as shown on the right of the slide. Now we will discuss the reaction between luminol and blood. Luminol combines with a basic solution, which removes the hydrogen protons from the nitrogen atoms. This results in the formation of two water molecules. This new molecule produced then reacts in order to move the negative formal charges from the nitrogen atoms to the oxygen atoms. Then, iron ions from the blood act as catalysts to decompose the hydrogen peroxide in the solution. Next, the nitrogen atoms are replaced with the oxygen atoms from the hydrogen peroxide molecule. This results in the production of a new molecule, as well as nitrogen gas. This process causes electrons to reach an excited state, which is very unstable, so the electrons return to their ground state. This process emits a photon, which allows light to be seen. The reaction mechanisms just described are shown here. You can see the movement of electrons as indicated by the curved arrows, and the photon produced at the end of the reaction is indicated by the red circle. Luminol's property of chemiluminescence is what makes it important in forensic science due to its ability to allow blood at crime scenes to be observed. When luminol is sprayed on blood, the iron in the blood catalyzes the reaction in the luminol solution. In the reaction described earlier, upon reaction with oxygen, an unstable organic peroxide is produced in an excited state. Due to this excited state, the molecule is extremely unstable. The electrons in this unstable excited state will transition to a stable ground state. This transition from the excited state to the ground state emits a photon of light as shown by the diagram on the left. This light is what is observed when luminol glows blue on top of blood. The infrared spectrum shows us one type of bond included in luminol structure. As indicated by the peak at nearly 3,000 wave numbers, we can tell that a molecule of luminol contains carbon to hydrogen bonds. We can also determine some of the connectivity of a luminol molecule by analyzing its H1 NMR spectrum. We can determine that luminol contains two singlets, two doublets, and one triplet. We can also use the locations of the peaks on the spectrum to figure out the relative distances between the hydrogen atoms and the electronegative atoms. I have ranked the hydrogens using the spectrum as shown on the left. Unfortunately, the use of luminol comes with a few potential disadvantages. It has been found to be catalyzed by substances other than blood. These include brain tissue, saliva, bleach, copper, rust, potassium permanganate, and bone marrow. Also, luminol can potentially damage or even destroy some genetic markers in cell DNA. Furthermore, luminol can smear blood, which causes prints and spatters to be harder to analyze. Besides its use in forensic science, luminol has other applications. In biology research, luminol is used to detect minimal amounts of DNA in proteins. In chemistry research, luminol is used to determine concentrations of metal ions. This helps with tracking reaction rates of oxidation in certain reactions. Furthermore, luminol is used commercially in glow sticks as well as in emergency light sources. So this concludes my discussion on the reaction mechanisms between luminol and blood, and I hope you now have a deeper understanding of luminol and its importance in everyday life. Thanks for watching.